Hello, everyone, and welcome back to the show. Today's guest is Jeremy Thomas. In today's episode, you can expect to learn how Duke University researchers are pushing the leading edge of brain healing, the history of stroke treatment methods, how and why a common skincare product has the same structure as the next generation of brain healing medicine, the next wave of potential advancements in Jeremy's field, and much more. Jeremy is a Biomedical Engineering PHD Candidate at Duke University, specifically in Tatiana Segura's lab. He has a bachelor's degree in biomedical engineering from Texas A&M, and he's passionate about ways to heal the brain after traumatic physical events. He's also been a friend of mine for many years. In an increasingly brain-optimized world, so much focus is going towards how to maximize all aspects of our brain function, whether that's through things like breathwork or supplements. At the same time, it feels like massive corporations are trying to hack into our brains through maximum dopamine response. It seems like the aftermath of severe brain injuries is almost going unnoticed or even being ignored. To me, it's healthy to bring research like Jeremy's into the spotlight so that we don't forget there are people struggling with these severe injuries. And it also reminds us that there's hope on the horizon for really all aspects of brain development and healing. Let's get into it. Jeremy, welcome to the show.
Hey.
So before we kind of dive into this whole stroke treatment thing, for people at home that don't know about that or don't know about you, give a little background on who you are, what you're kind of, I'm going to call it an expertise, relative to me at least, and what that lies in, what you do, and what the why kind of is behind that.
Sure. Yeah. I'm happy to be here. My name's Jeremy Thomas. I'm a third year PhD at Duke University. It's around halfway through my program. In my program, I do research in a lab that develops materials for stroke. So we focus on treating stroke by developing these biomaterials that are composed of these tiny spheres all attached to each other, which I'm sure we'll get into more later. We can inject that material into where a stroke occurs, and that kind of provides a scaffold for cells all around where the stroke is to kind of grow in and repair the damaged area. The hope is that this can eventually help people recover from strokes more effectively. And my why for this, I got into research into like this biomedical engineering field of research early on in undergrad, and just decided that it seemed like a really impactful and cool way to help people. And so, I decided to go down this PhD route, this like research heavy route, because of that. And as far as why we do our research, we want to help people recover from strokes, and even some other diseases and things that our lab focuses on. So really excited to talk about it all today.
Awesome, yeah. Could you give like maybe a little bit of an insight into the history of stroke treatment and stroke research? Just give us a kind of a baseline, a foothold to start off of.
Yeah, definitely. So, to begin, there are really a couple types of stroke. Um, one type of stroke is called a hemorrhagic stroke, and that's where there's just a massive blood loss in one area of the brain, usually from like a big trauma, like hitting your head or something. Um, and that big massive blood loss means one area of your brain doesn't receive the nutrients it needs. So that area of the brain begins to die pretty much. Your blood carries nutrients all over your body, and so when an area doesn't receive those nutrients, it really doesn't like it, and a stroke is that happening in the brain. That accounts for around 20% of strokes. The other more common type of stroke, which is really what our treatment targets, is called ischemic stroke. And so that's where you have a blood clot in the area of your brain. So instead of losing a lot of blood is just kind of blocked. Blood flow is blocked to one area of the brain, causing that same tissue death. And so that's around 80% of strokes. So that's where the majority of treatment kind of targets. And so, to go down a little further, when you have a stroke, there are a few phases of stroke recovery. The immediate phase is called the acute phase. and that's where kind of all of that tissue death is happening. And that's where the majority of stroke treatments have targeted, which makes sense. That's the most immediate way to help people live and also recover from their stroke. So there are some treatments where you can break up that blood clot or like kind of grab the blood clot and take it out. Pretty crazy stuff to me. Um, and uh, those treatments really became big and widespread in like the 90s, and they really helped drive stroke mortality down a lot, which is amazing. I mean, awesome. But the issue is like a little over half of people that have a stroke have a long-term disability. And there really aren't a lot of things besides just like physical rehabilitation, like just physical rehab to help people with those long-term physical disabilities. And so that's where the next phase of stroke recovery comes in. It's called the subacute phase, the next one. And in that phase, the rest of the brain sort of tries to take over whichever part of the brain had that damage. So one area has the damage, the area right around it is like, well, where did that go? Okay, I'm going to try and take over what that part of the brain used to like specialize in. So you see like a big wave in what's called neuroplasticity, which is where all the cells around there trying to sort of remake their connections to kind of take over that function. So a lot of the current treatments, they target that subacute phase to try and either lengthen that neuroplasticity or just make it more intense so that people will have less disability long term. So instead of the fixing the mortality part, it's more about trying to fix the long term disability. And that's where our material and a lot of the current newer treatments come in.
Wow, that is fascinating. I had no idea that it was genuine neuroplasticity. I thought that it was kind of just a like, trying to catch up with
Mm-hmm.
the part that was harmed. Um, that's really cool, and they're trying to like kind of get in there in that time period. And I guess would it be the right characterization to say that you're trying to help that time period be more effective, essentially?
Mm-hmm. Yeah, yeah, I think that's a great way to say it. Um, it's really interesting. Things like neuroplasticity, uh, they're really associated with like early life. When someone's just born, or even they're still like, within like in utero, like still developing and as like a fetus or whatever. Um, that's when you talk about things like neuroplasticity, or like new cells being formed, like from stem cells and things. But what's interesting is in a lot of when an injury happens in the body, you often see a lot of those same mechanisms come back, even though you're an adult and they aren't happening as intensely or as frequently. And so neuroplasticity is one example of that, where your brain's always a little neuroplastic, but when there's an injury, those like pathways and mechanisms, they kind of sprout up again. And so that's what a lot of these therapies try to take advantage of.
You used a term there, in utero. You kind of already defined what a stroke is, but could you just kind of, quickly define a little baseline for what like in vitro, in vivo, maybe a few
Mhm.
other key terms that'll help us understand what you're working on?
Yeah, sure. So, in vitro is a term commonly used in scientific research where you're doing experiments without like a living organism, sort of. So it's more with cells. So instead of doing something in like an animal model or doing some kind of human research, you're doing things just in cells, and that can be really useful because you can answer some really specific questions. Like if I wanted to look at a specific cell type and say, how does this specific cell type respond to this material we're making? It'd be really hard to do that in like if you had a full brain and you were looking at things because there's so many cells and so many things happening. So in vitro experiments are just kind of with only cells. I think that's the best way to think about it. And in vivo is just similar, except it's in like a living organism. So in an animal model or in like a human model if you were to do that, or a large animal model or something like that.
Cool. Just kind of linger on that for a quick second.
Mhm.
Um, how do you know whenever you're doing research that you're kind of ready to go from an in vitro model to an in vivo
Mhm.
model?
Yeah, so this people debate this a lot, and people have different viewpoints on this. Um, like some for our material, for example, uh, we validated a while ago that it's safe in animals and like in organisms. Um, and so because of that, it's not too uncommon for us we make like a small tweak, and then we can just go straight to the in vivo, the straight to the animal model stuff, because it's safe, it's not going to hurt the thing, and that information is going to be most relevant to like the next stage, the clinical trial stage, the seeing if it's going to actually be beneficial to humans. And so for us, it's a lot about the question we're trying to answer. So like if it's a more specific question about a specific cell type, then maybe you want to start in vitro. If you want to see, hey, is this thing going to be really beneficial for treating stroke? Is this thing going to help people recover from stroke? You go to the animal model. I think when it's a newer therapy, you might start more at the in vitro level, just for seeing if it's a super new, like, hey, is this going to like hurt cells? We don't want to put something in an animal that's going to like not help it, and be like toxic or whatever. Then you'd start in the cells and then move to the animal model. Yeah.
So you're kind of setting up your own kind of boundaries of where you can work within whatever you're trying to test.
Yeah, and I think that's there's within academia, I guess, and scientific research in general, there's bioethicists that play a role. So they're into ethics, and there's a huge arm over our university that focuses only on animal ethics and animal safety, which anything that you ever want to do in an animal, you have to write up a very detailed protocol, send it to them, they review it, they tell you, hey, don't forget about this, to make sure the animal doesn't feel pain or any of those things, or to minimize all of those things. They send it back, and then that's what you have to kind of operate under those bounds. So I guess that's kind of how we get our bounds. But like once we have that protocol for our material, if you make a small tweak to the material, you can just go right back into the animal model.
Because the groundwork's already been laid for that. That makes sense.
Yeah.
So, okay, there's a little nuance in that, I think. Whenever you're looking at wound healing, and specifically with stroke healing, how do you test wound healing in that kind of framework of the animal ideally not feeling pain, or like within that like ethics boundary
Yeah.
that you're being given?
Yeah. So the wound healing animal models. So I mentioned I'm on the stroke side, but our lab also has a background in skin wound healing. Skin wounds, when they heal with like a scar or anything like that, it decreases the strength of the wound, which is not good for preventing like bad things from entering your body, preventing re-injury, things like that. So this material can also be injected in the skin wounds to kind of help the wound recover with more strength, and even more quickly. And that's actually a little further along in our lab. So that material is kind of in clinical trials now. But so I'm not too familiar with their animal model, and how they, a lot of times they'll recommend like make sure you use this kind of what's it called? Not a painkiller, but something to minimize pain.
Like an anesthetic?
Yes, yeah, yeah. Things like that are incorporated into protocols to make sure that the animal doesn't feel pain. Like I know for our stroke protocol, the areas that we are making incisions and things for surgeries happen to not have a lot of like dense nerves, sort of, for the mouse. Yeah, we use mice as our main animal model, and so that is in and of itself kind of minimizes animal pain. But I think there's just kind of a threshold that the animal ethics people agree is reasonable, and so they just make sure that people are administering what they need to minimize pain with their protocols.
Fair enough. I trust they know what they're talking about. So, what is this material that you're working with? And like, what's kind of I don't know if there's some kind of secret sauce in there, but like, what is it? What's the base framework of it? And what I want to eventually get to is why does it work so well? Because this is pretty fascinating. These papers you sent me are maybe a little bit above my head, but I get the gist of it, and this is pretty cool stuff.
Yeah, it's pretty crazy. So our material, the acronym we use for it is MAP. And so that stands for microporous annealed particle. So MAP scaffolds, microporous annealed particle scaffolds. And so what they are, we make these tiny spheres. They're like 100 micrometers long, so really small. Um, these tiny spheres, and they're made of hydrogel. Um, Hydrogel is like it's really similar to Jello. Like it's like a gelatin is even like a common base for a hydrogel. We don't use it, but that's a common one. They're like these Jello spheres, pretty much, and we make them so that they can attach to each other. We add these other annealing agents. They cause chemical reaction to occur, so everything attaches, and so then you end up with this scaffold of a bunch of spheres attached to each other. Um, and if you to help like imagine what that looks like, if you imagine like a gumball machine, like a bunch of gumballs like sitting in the top, that's kind of how it is. Like a bunch of spheres all kind of packed together as close as they can be. And if you think about that gumball machine, you think there's a lot of space in there. Like they can't, it's not like if you had a bunch of rectangular blocks, they could fit in perfectly with no space between. The spheres, a bunch of spheres together, there's a bunch of pores, like a bunch of space in between. And so that space alone is sort of the secret sauce of why this material works different from other materials, and kind of a big benefit it has is that's also the M, microporous. We've got these micropores in there, and um, the any surrounding tissue. So like if it's a skin wound, all the surrounding cells. If it's a stroke, all the surrounding cells in the brain. Because of that space, they're able to grow into the scaffold really easily. We can also attach things to those spheres that will make the cell be like, hey, I want to grow in there. Look at that thing. And then they'll kind of grow towards it, things like that, things that allow the cells to grab onto our spheres really easily. And, I guess to give a little more context on why this is like an improvement over what's been before, hydrogels, the like jello type materials, those are really common for a lot of like our biomedical engineering, like promoting regeneration, a lot of this field. But in the past, they were typically just like kind of blocks of hydrogel, like a block of jello, and you would kind of put that in a wound, and it would provide structure because instead of air, there's something there, and then the surrounding cells can kind of degrade that hydrogel and kind of break into it. But it's harder, and so it's not as easy for the surrounding cells to sort of break in there. And so that's kind of where our map scaffolds are, like spherical, granular scaffolds. Um, that's sort of their secret sauce, I think, is their porosity.
Okay, so it's a big improvement over previous maybe types, or maybe not even types, but just previous iterations of this because it's injectable, or what makes this like particularly unique as a
Hmm.
new substance?
Yeah, so that's another part of it. So I mentioned if you have a block of Jello, you can't really like inject that anywhere. Like you can't suck it up into a thing to inject most of the time unless it's got like a really particular property that allows that.
It's
Um,
hard to put a block of Jello into the brain.
exactly, exactly. Yeah, and so this one, these spheres, even though if you took one sphere, it's similar to a block, like it's a solid thing. Since uh, they have to have some space between them. They're injectable, so you can. The way our stroke stuff works, there's a stroke. You load a syringe with all of those kind of spheres all packed together. Um, right before you inject them into the brain, you add the thing that's going to allow them to attach to each other. That doesn't happen instantly. It takes some time, and so sort of as that connection is happening, you inject into where the stroke is. I didn't really mention it earlier, but when there's a stroke, uh, it creates kind of a nice pocket for something to be injected in. Like all of that dead and dying tissue, it's still there, but it's not solid brain. Like it's not a hole, but it's almost like it's called the infarct. It's like a little area that you can inject something into. Um, so you inject all the spheres that are in the process of connecting to each other into the brain, and then sort of in that spot they all connect to make what's similar to a block, honestly. Like once the spheres all connect to each other, it's almost like a block, like it's a solid piece of jello effectively. But if you were to zoom in on that block, there's a ton of space in there that allows the cells to grow. And our the earlier papers from this lab before I joined show that without attaching anything crazy to the spheres, even just having that structure alone in there, especially for the skin wounds, promotes regeneration. Like just the micropore structure with like one thing attached to allow the cells to grab on to the sphere, that alone promotes regeneration. And you can take it further by attaching even more like pro repair things to the spheres too.
I remember reading through, maybe it was the second one you sent me. It was something like over 10% more effective than like what the previous. I don't know if you can call it standard, but what the previous kind of iteration was. That's that's statistically significant.
Hmm.
It was like 39% wound regeneration as opposed to like 28 or 27, something like that. That's a stunning improvement, just from that structure you're talking about.
Yeah,
So talk to me about that a little bit.
yeah, it's pretty crazy. The human body and like all animals' body, like it's pretty crazy how responsive they are to things. Even just having a block of a thing nearby with kind of a crazy structure, all the cells in there can kind of respond to that without even having anything else in there that's super like crazy or biologically active. Um, and so, it is pretty wild, I think, and particularly in the skin wounds you see with that structure alone, tons of growth. Um, and one unique part of these spheres is, that they attach to each other through these functional handles. There's a ton of chemistry that you can get into with them to attach even more things. And so, in more recent work, our lab has looked at, well, what if we attach this particular thing that promotes stroke regeneration, like just in the natural pathway of stroke regeneration. Well, then we see even better responses. What if we attach uh, this like sugar that is just part of a lot of repair processes throughout the body and is pretty present in the brain, even more repair. And so the uh, the scaffold, that's another benefit to it, is it has that structure that allows for tissue growth, um, but it can also kind of act as like a delivery vehicle, or like you can also deliver things to the either the wound area or the stroke area by attaching more things to those spheres.
Do you think that stroke treatment is a kind of a natural next step? I mean, obviously it's already in process, but like from that initial like skin wound research, is stroke treatment kind of a natural following step, or is this kind of or was it kind of a big leap, and now we're realizing, oh, this is kind of amazing?
Yeah, that's a great question because when I first joined the lab, I was like skin versus brain. Like those are almost like, I don't know, at least the way I thought about it coming in, they're almost like opposites just in terms of well, the brain's pretty unique to most other things in the body, I would say. So, um, I think this is can kind of get into just the way um, science researchers and like engineering professors in academia and all these people think about how they run their labs and how they sort of take next steps with their technologies. Um, they look for gaps usually, and so there are other ways that this material can be applied. Like we've had students who are either students or they're postdocs, which means they had their PhD but they were working in our lab, and they went on to form labs that are focusing on other areas. Like I think one person's maybe using this material for like pancreas things for diabetes. I think. Like using it as like a delivery vehicle for insulin or something like that. I'm not super familiar, but I think my assumption is that when our lab was looking at where to apply this material next, they were looking for just something that needed uh, stronger therapies and something that maybe biomaterials weren't applied to a lot yet. And so I think stroke was that answer. Another thing uh, that I've kind of realized as I've been around here a bit more is you can often make your decisions just based on who happens to be around you. And what I mean by that is, when this first started, this lab was actually at UCLA. They moved like 5 or 6 years ago to Duke. And they had a collaborator who was really, really big, a really, really big neurologist, big stroke expert. And so he had done some, um, maybe he had already done a little bit of materials things with stroke, but not with our material. And so I think that I don't know this for sure, but I would assume that was part of the motivation too, is that we have this collaborator here, and this person is an expert. This could happen, and this could be great. So it's not always that we have the infinite number of possibilities for where to apply the technology. It can also be that, we could apply it in these few different ways. This one, there's an expert here, and they already have some expertise, and like we could collaborate with them, and I think that would be a good step for us.
What a great explanation of like how scientific curiosity works. It's
It's
like
pretty
you want
crazy.
something both new and like novel, and also um, maybe this is the wrong word, but like kind of convenient, like the
Mhm.
convenience of someone being right there that is an expert in this field, and then you have this tissue or a material that's never been used. It's like just looking for where those new connections are to be
Mhm.
made.
Yeah, yeah, it's less cut and dry than I first thought coming in about like, okay, the very best next best thing is this, and so we're going to do this, because science it's really hard. It takes like a really, really long time to do anything. And so having things like a strong collaborator, um, the convenience factor, like that's a big part of it.
So in this kind of world that you're in, could you explain maybe the difference between your microporous material and how that's different than maybe just this might be the wrong phrasing, but like just a maybe traditionally porous material versus even a non-porous
Yeah,
material? What does that look
yeah,
like?
yeah, great question. Um, so typically when you talk about a non-porous material, and I'll keep it in the context of hydrogels because that's what I'm most familiar with. Uh,
Mhm.
that would just be kind of like that Jello block. So that would be considered non-porous. If you really zoom in on the Jello block, there's sort of a mesh of polymer happening in there, and that's the case with most solids, I guess. Um, but there that people tend to refer to as like more of a mesh than as porosity. Um, and so you have a block of this jello thing. When you zoom in, there's like strands of polymer, kind of. They're really tiny. You can't like really see them like that. Um, but what people who have really focused on those materials have done is you can impact how big this mesh is, how big or small the mesh is. So there are people who have shown that uh, when you make this mesh sort of larger, then if you have something in that hydrogel, it'll release faster. And so they can control like release rates of things with that. Um, the porous hydrogel versus like our granular microporous thing, people have taken those like non-porous hydrogels and they've made pores in them through like a few different ways. Just like when you make the hydrogel, you can have I think they call them porogens, maybe. Like you can have things in there that you're able to take out after the gel is formed. So if you think about, uh, I don't know, like you have a block of gel that's kind of liquidy before the gel is actually made. If you stuck like a pencil through the top of it or something, and then the gel formed, and then you took the pencil out, there'd be a big hole like in the top. It's like that, except not a pencil, and like these tiny porogens. And um, so they would often do things like that. And there are a lot of ways they would make these pores. Like that's one. Um, the micropores thing, one reason why that's beneficial is you can really control the consistency and like characteristics of the pores. So, um, ours, it's instead of uh, having a block and making like pores, we have the circles, we're attaching them together, and that naturally has um, some intrinsic characteristics, I guess. We also have this software that a previous PhD student made. It's called Lobamap, and through that they can simulate a bunch of different scaffolds and then make judgments on what the pores in those scaffolds look like to kind of inform what we do. And so they can say like, okay, if you make your spheres this big, you're going to get this many of this type of pore, and like the pores are going to look like this. The pores will be this big, this small, whatever. Um, and So you just have more control. Like we can control how well things are packed before we inject them, and like how well they're packed. That impacts how the pores look, and how the cells can like behave with the pores, I guess.
Okay, so to make sure that I kind of am understanding the like the microporous, like the very granular type, there's just genuinely a higher level of control than like a traditional porous type
Mhm.
one where you're kind of having placeholders that get removed or various other techniques. There's just a higher level of precision. Is that what it is?
That's a big part of it. And I think one other thing that I forgot to say is that there's kind of a more guaranteed porous network in the microporous one. Like the pores are
Hmm.
all connected to each other because like you think gumballs, like all there are paths there. And whereas if you just make some pores in a block of hydrogel, they aren't necessarily connected. There might be some pores that things can get into, but not every pore is connected to each other necessarily. Um, and so that's another benefit that I think the like our map material offers.
Yeah, why is that such a big benefit to have them very connected to each other like that?
Yeah, so if you think about like a cell or a bunch of cells trying to grow into a scaffold, Um, the reason why it's difficult for, like, if you have a block of gel with no pores, why that's hard is because when a cell hits a wall, these hydrogels are designed to where the cells can kind of eat into them a little bit, like they're made that way. The cells and like other things that are with the cells can break through the hydrogel and grow into it. But that's much more difficult than if it's just kind of an open space for the cell to grow into, and there's maybe like hydrogel along the boundary that can it can crawl, like kind of grab onto to crawl through. So if you have an interconnected, it's kind of like if you were to go into like a cave or something, and like you hit a wall in a cave. To go any further, you have to like dig through that wall. Like you'd have to have some TNT, like whatever, to break through the wall. If it was just open air the whole time, then you're just walking through the cave, you're fine. It's much easier, and it works the same way. Like when things are interconnected, you have a network of pores. The cells can easily grow through everything. If you have just some pores but they aren't connected, you'll hit some walls along the way, which can be difficult.
Okay, so you can use the kind of pathways of the porosity to probably store some medications and things like that and other
Mm-hmm.
things that assist in the healing, like you were saying. But it
Mm-hmm.
also, like using the cave analogy, it like the cave itself isn't what you're like the walls of the cave is not what you're going into. You're going through the cave, and the cave guides you where maybe it's helpful to go. And so for the hydrogels, that following that same logic, you're basically just like guiding the new cells to grow further and more effectively. Is that how it is?
Yeah. That's a big part of it. Um, yeah, to extend the analogy, like you're walking on the floor of the cave. The cave like provides structure for you to be able to walk. The hydrogels, we coat them. It's we typically use what's called RGD. It's like a cell adhesion thing, and so because that's there, the cells are able to like grab onto something. Like they're even referred to as like binding handles. Sometimes like you can imagine a cell like it doesn't look like that, but it's like climbing along the hydrogel because those things are there. So like if those weren't there, the cell couldn't really go through. Uh, so yeah, it like you said, it can act as like a sustained delivery vehicle for reparative things, and it can act as like a almost like a handlebar for cells to crawl through the scaffold.
Hmm. So give me kind of an overview of some of the research maybe you're working on now in comparison with previous uh, papers that have been put together, and kind of how this effectiveness has been improving or growing and what you're doing in your daily work.
Yeah, sure. So I'm currently to give more context on like how these PhD programs work and everything. I'm starting my third year, and so around this time, people do what's called a preliminary exam. And your preliminary exam, and this works different at all different programs, but like at our program, you take this preliminary exam and you say what you want to work on for your thesis. You propose a bunch of project ideas. Maybe you've done a little bit of them, but most of them are you haven't done them yet. And then there are a few professors, and they give you like feedback and say, that won't really work. This will work. Don't forget to consider this. And um, from after that, that's when you really get going on your stuff. So I'm kind of in that stage right now. So I can't comment too much on how well things will work. Hopefully they work super well. If they don't, I'll have to pivot and do something else, which is always stressful, but that's part of this.
Mhm.
My thesis stuff right now is kind of focuses on two like two different goals. So the first is to be able to optimize our materials more quickly. And so it it's kind of laborious to make one of these due to a bunch of different things. It's not you can't make these like scaffolds super quickly. And so if you have like five different scaffolds that you wanted to try because you would think that maybe they would all work well in different ways. Um, excuse me. Um, you could really only pick like one or two to focus on for someone's PhD just because they take forever, not forever, but like it takes a lot of labor to make them. One other thing is that, I mentioned that these scaffolds can act as delivery vehicles. One thing that we started looking at more is you can maybe take three different types of these spheres, and maybe this one's really good for promoting vessel formation, this one's good for something else, whatever. And you can mix them together and have like a scaffold with a third of each one to effectively deliver like three different materials at once. That's great. Preparing those takes even more time. And uh, if you wanted to compare, like, what's the ideal ratio, you can just imagine how many samples, like, it would just take your experiments just become massive. Instead of having like a dozen or like a dozen at like 18 scaffolds for one in vitro like cell experiment, you'd want to have like close to 100. And that's just not possible with our techniques right now. And so I'm trying to do some things to uh, to speed up that process, like taking advantage of some robotics in our department to maybe like do some pipetting for us or some movement of the microparticles for us. I'm trying to do some things. Right now, we often will have to take images with a microscope if we're doing our cell stuff and like look at, hey, how well do these vessels form? Let me look at it under the microscope and then look at each individual scaffold, go in, quantify, like, look at how long the vessels are, like how well they're formed, how many branches do they form, things like that. But that takes a super long time. And so I'm also trying to come up with some new ways for us to even know if the cells in the material like worked and looked preparative. So that's the kind of the one main goal here. The other one is to uh, look at our materials with some other novel therapies that are targeting. I mentioned the subacute phase earlier, like the phase with all the neuroplasticity. And we're not the only people doing that. Like there are people that are trying to do like different types of stimulation. So like electrical stimulation, magnetic stimulation to do some different things. People are trying to do, I think, like some stem cell injection things too. Um, and so trying to combine our material with one of those to see if maybe the combination of them leads to some really cool recovery results.
Wow, that is a lot of things. How do you like balance doing what seems to me like three different projects into kind of one thesis?
Yeah, so that's a great question. That's also a comment my committee gave me. So, um, so the reason why and this varies by like different professors have different like thoughts on how to structure people's PhDs. I think mine kind of she likes for us to have sort of a wider scope at the beginning because things will inevitably not work. Um, that's just how it always goes. And so, I have these three things. It is kind of required for your thesis work to have three aims and for each aim to be distinct from the other. They are, I think, normally a little more connected. So, like, for example, maybe you'd have three in that making things more optimized camp, or like three in the combining the therapies camp. Um, but it is sort of required that they don't depend on each other. That's sort of like if aim two depends on aim one, they don't like that because then if aim one fails, then like two-thirds of your thesis is like out the window, and that's scary and stressful.
They're almost kind of the same aim if they're dependent.
Yes, exactly. And like they do some like funding things too. Ideally, this work will
Hmm.
be fundable from the National Institute of Health, or whoever's funding our lab. And those people also require for AIMS to be independent for the same reason. Like they don't want to fund a big project. One part of it doesn't work early on, then they give all this money to a lab and then none of it works. Um, and so I guess as far as how to balance that, it's not just in scientific research, but like fail fast is a common like mindset with this stuff. And so you try to early on have some experiments that if they that are like no go. If they work, then you keep going, and if they don't, then you can just abandon the thing early, so you don't spend like a really long time on something that's going to inevitably fail. Um, and so yeah, that's probably the main thing.
It's like experimental design becomes very important.
Hmm.
So yeah, okay, yeah, that makes a lot of sense. So what kind of time frame are you looking at whenever you say these take a long time to make and you're wanting to make that a little more expedient? Like how long do they take to make? And um, I'm sure maybe you don't have a goal in mind, but like what kind of improvement do you think there even could be?
Yeah, that's a great question. So it's not like insanely long. It's long relative to bulk hydrogel, like other biomaterials, like the non-porous guys. So to make these spheres, we make them through microfluidics. And so what that is, it's these like little rectangular blocks of a different material. And then there's these like tiny channels, like tiny tubes, and they're micro size. Like, so that's why it's called microfluidics. Um, and you can make these spheres through a bunch of different ways, but you flow your um, the liquid that's going to eventually be your like jello spheres through this chip. We call them chips, microfluidic chips. These blocks of rectangular stuff with all the channels in there. Um, Work in a bunch of different ways, but eventually they make some spheres, and they come out the other end as these tiny, like perfectly, ideally like perfectly, they look the same, identical spheres. And so that takes some time, and it depends a lot on what you're making your spheres out of. But there's actually been some new like innovations recently that's made that much faster. But you can make with this even with these new innovations, you're making on like the single milliliters of spheres um, per hour, pretty much. Maybe in like the tens of milliliters of spheres per hour. And so for like our animal models, that's okay. Like those only require like 6 microliters to treat one stroke, like not very much. Um, and but like in a human equivalent thing, a larger stroke, I don't know the exact volume, but like larger strokes can be much larger than that. Like we currently to make enough material for like a large stroke, um, that would not be super feasible. And so that takes a lot of time, uh, sort of, depending on how you're talking about things. For like a typical,
And
go ahead.
especially when you're talking about like repeatability of experiments,
Hmm.
you're talking many, many, many hours to be able to have enough. It seems to be able to have, like you said, what could be on the order of 100 different
Yep.
experimental directions you need to go.
Yeah, yeah, exactly. Yeah, so like for our in vitro stuff, when we're doing our cell experiments, that volume, part of making this faster is I'm trying to find some methods where we use less volume per sample. Before it was, we would often have like 50 microliters per sample, and you want 3 samples per group, 3 samples per like, condition you're trying to look at. And so that's more than like the animal models. Like the animal models is 6 microliters per animal. This is 50 microliters, really 150 microliters per like type of material you're trying to test or whatever. And so that can balloon. Like if you're getting close to 100, that's like 5 milliliters of stuff right there. And so, um, so that's part of it. The other part that makes things slow, typically when you're doing science, you have to use these micropipettes, um, which are, I don't know how to really describe. pipettes, but they just like suck up a set amount of liquid. So like you'll tell the thing, I want to suck up 100 microliters of liquid. It's super precise. It'll suck up exactly 100 microliters of liquid. Um, when you're doing large studies for like developing new uh, drugs or whatever, like new those kind of things, they'll have these like lines of pipettes, like 12 of them in a row, so you can go really fast through a bunch of things. That doesn't work for our material. Our material, while it is injectable and pipettable, it isn't injectable and pipettable as these just pure liquids. It's a little more viscous. It's a little thicker. And so we have to use a specific type of pipette to move them. And those specific type of pipettes, they don't have the like multi-channel. So you have to go one at a time through everything. So if you had like nearly 100, let's say you wanted to make like 99 scaffolds with different ratios of 3 different types of material, you maybe start with like 33 of them or something, and then like start doing like things like that. It would just take forever. Super prone to have a tiny error and you not realize it. All of this, like all of these things, and so um, that's really laborious and uh, just also discourages people from wanting to do larger screens. The last thing to consider with that, cells don't stay alive forever when they aren't like in a some nice conditions and like the liquid they like and the like body temperature and perfect amounts of like CO2 regulated. And so as we're doing when we put our cells inside of our material, they're sitting out for a second, like as they're getting mixed in. As you increase the number of materials you have, like if you're near 100, the cells would be sitting out for a while, they would start to die. And that also impacts your results, like, and your ability to do larger screens. Um, and so that also increases labor and the need for things to be faster. Like we need things to get mixed together faster so that cells don't die. That's another part of it.
Mm. So really, like that almost seems like could potentially be a backbone of making all of the research of these materials and this treatment more effective moving forward.
Mhm.
That is really cool. I
It'd
think
be
that's
really
like,
cool. It'd be cool if it worked. I mean, the robotics, they're really common in like drug discovery research. So that's like, big pharma companies, they'll have like these automated robotic systems where they'll say like, we want a drug that like targets this particular receptor or whatever. We in our library, because we're a big pharma company and we have lots of money, we have millions of like potential drugs that could potentially target this thing. So they have these robotics cycle through millions and millions of things like like fast, and so that technology exists. It's never been applied to biomaterials because most biomaterials are not pipettable. And so that's a unique thing with our stuff in that uh, they can almost act like a typical drug discovery type research reagent. They're not, we'd have to tune it to make sure things could be pipetted by the robots, but um, it's kind of a newer thing with our like more microporous granular hydrogels.
yeah, if I was on your committee, I would tell you that seems like a perfect novel, but potentially feasible thesis topic.
Thanks,
But
thanks. Yeah, I hope it works out.
yeah, so you mentioned the kind of the third leg of your potential thesis topics about integration with other technologies.
Mhm.
That is super interesting to me, and I'm curious where you kind of see the main opportunities there, because obviously with this material itself seeming to work so well. What are some of the other like technologies that for treatments that exist now? Maybe you've even had clinical trials because they're less or non-invasive, and how do they kind of interplay with what you're working on?
Yeah, so, you know, one of the things that motivated me to like start looking at some of these things is academic research can get really siloed. And there are pros and cons to the way that we do like academic engineering research, but it's very like patent driven, like intellectual property, IP stuff. Like people, they want to develop something that will be patented. They can take that patent and make a startup out of it or whatever.
Yeah.
And there are pros and cons to that. Like, one pro, that kind of stuff can be motivating to a lot of people. It can encourage a lot of like it can encourage private interest in funding things like when it's around the clinical trial stage. One con is it doesn't really uh, encourage collaboration all that often. Um, and so like and I'll talk about a couple of them in a second, like there are other great technologies that target the subacute phase of stroke recovery, but it's not always advantageous for people to think about how they can combine their things because they would rather stick with what they have because they have their patents around it and whatever, and then move forward. Luckily, my professor doesn't really follow that, and this person we're collaborating with doesn't really follow that to that level at least, and so we uh, I guess the one that we're going to try and do is more in like the stimulation realm, and so um, like I'm interested, or sorry, not I'm interested. The field has tried a few different types of stimulation therapies for treating stroke, and by stimulation that can be really anything. There's a ton of different things that would kind of stimulation, but I'm talking more about like uh, magnetic or electrical stimulation, like really low levels of that, and so there's both non-invasive and invasive categories like under this type of thing. And so like you can have a non-invasive thing where there's like some magnetic stimulation kind of just, or electrical sort of up here. That can really impact how the different brain hemispheres talk to each other. So they can control, I think, I guess post-stroke, there's a lot of like instability in how they talk to each other. And so that can kind of stabilize things a little bit. Um, there's also the more invasive stimulation therapies. So there's like deep brain stimulation, which kind of like injects something and tries to stimulate like a very specific part of the brain to do it for a particular purpose. There's cortical stimulation, which is more what we're looking at. That's where you have like a kind of stimulation area right on the motor cortex, like right on top of where, for us, that's where we apply our strokes usually for our animal models. So like right on top of where the stroke is. The goal of that is to encourage the neurons to form their new connections, like the neuroplasticity part of things. Um, uh, and in theory, that's really compatible with our material. Like you could inject the material and then put the stimulation stuff on top of that, or vice versa in the order. And they could kind of work together. You could even, if that worked, you could go a step further and like change the properties of the material to where it could take advantage of the stimulation there. So that's really what my project is. But there are a few other subacute therapies, but I think that's the main thing we're trying to do.
Yeah, kind of the main options that seem feasible, probably with kind of the direction you wanted to go.
Yeah, and part of that is that it goes back to the collaborator thing too. So we like at where I am, like there are people who do all of those different types of stimulation therapies, um, but they're all great. They all would have been great, and the person we're working with is awesome. But we worked with the person that my professor already knew well and had worked with. I don't know if they worked together before, but like knew pretty well, knew that he was like a good person and would be a good collaborator. And he had more expertise in the invasive stimulation and was really interested in the cortical stimulation. And so that's what we're going with. I honestly, in theory, all of these could be beneficial together, but that's another example of like proximity and just like who happens to know each other sparking like a particular avenue. And you also mentioned a little bit about clinical trials. So this stimulation stuff, it has a really actually interesting history in clinical trials. The cortical stimulation in particular, around like 2010, was looking really promising for treating stroke, like what we're trying to do like right now, 15 years later. And this startup had a bunch of funding. All of their like animal stuff was looking amazing. They went to clinical trial. Clinical trial is divided in like 3 phases typically. First phase is looking at like, is the thing safe? Not even focusing that much on how well it works. Like, is it safe? Phase 2 looks more at the results. Phase 3 is similar to phase 2, but like way more people, just huge, just massive. And So phase 1 and 2 looked amazing. They were really excited. Phase 3 tanked. And so the company like the value of the company went down like 1% of what it was, and then they sold, and it was just so since then everybody was like, no, we're not trying that again. Like that was scary. That was like there you can find press articles from the time that are like this wasn't just a failure for this company. This is a failure for like the entire field of like just very dramatic. And so I think for that reason there was some hesitation. But since the results were so promising, people are coming back to it. And
Hmm.
people have also often speculated that, well, and side note, that's really good motivation for combining that with another therapy that works well. Maybe you just need a little more to get over the hump, like that kind of thing. Um,
Hmm.
but a lot of people speculate that the reason that phase 3 trial failed was that was more about like patient selection than the actual therapy. Um, the the way that they decide at what level to stimulate someone, like what current value to like how strong to provide the stimulation to a patient is you start at a low level and kind of increase until you see like a small response and like that the hand opposite of whatever brain side you're stimulating. And but if someone's stroke is severe enough that pathway is damaged and so you can't see a response in the hand. And so I think what they did was whoever they could get that value from, they would kind of average that out for the people who they couldn't see that like motor response from. Um, in phase 1 and 2, they could see a motor response from like 80% of the people that they enlisted in the trial. Phase 3, I think since it was larger and they needed more people, it was like 15% of the people they could see a response from. And so the speculation is that I think early on they were like, well, we think it still works. We'll just have to average out the value. I think the speculation afterwards was, well, maybe this is really only beneficial if that pathway is still connected. Maybe, and so they looked at only the 15% of people who had that response, and they did see it like a significant increase in their functional recovery in that phase 3 trial. But if you like threw it all together with everybody, they didn't. And so it's kind of interesting how really small things like that can like completely change what's available to patients and like what makes it through clinical trials.
Hmm. Yeah, and what you just said specifically, actually, at least to me, with my very little knowledge of really the details behind this, that is like exciting to me. With that, okay, you see, whenever you have like kind of what you're saying, they postulated afterwards is that okay, maybe you have to have that connection be somewhat viable still.
Mhm.
Well, that looks like a big opportunity, like you're saying, to kind of work together and maybe bridge that gap with the injectable, where you have something local that promotes the healing and the vasculature, and then the external, like electrical stimulation or like I would whatever the
Yeah.
electrical stimulation is, internal or external, together. That seemed like a really huge, like novel opportunity, to even my uneducated perspective on it.
No, I think it would be cool. I hope it works. It can be hard to, we work in mice, and so it can be hard to, right now we're just working on making the model. And so for all of these things, for you have to start in animals because you can't just start stimulating a human with like you have to start somewhere before that. There's the way our current system is. I think ideally, just a quick note on like animal models. I think ideally the field eventually will have no need for animals. There's a lot of technology being worked on to replace animal, like the microfluidic stuff I mentioned. There are people working on developing these like labs on a chip, is what they call them, as ways to simulate complex cellular responses and cellular interactions to try and get things that um, to not have to use animal models as much. Um, Honestly, I don't think in our lifetime technology will develop enough to have no use for them, but like eventually that would be a cool thing. But right now, you start with trying to develop a model that allows injection of the material with stimulation so that you can even answer the questions you want to answer. And that's where we are now.
Kind of taking a bit of a step back a little bit. something that I've kind of thought about as we've been talking about this is we've used the word hydrogel a lot, and kind of just a funny connection that I've made is you see the word hydrogel in a lot of skincare products. Is
Mhm.
this kind of the same avenue as that?
Yes, yeah, and I honestly don't know a lot about the skincare products that, uh, but yeah, there's a lot of crossover. Like the main polymer we use for our materials is hyaluronic acid, which you see hyaluronic acid on so
Oh,
many products. Um,
yeah.
and I don't know a lot about even like how skincare products like work, like how they actually work, to be honest. But I think, a lot of the things we use are from like the extracellular matrix throughout our body. And so like we use components that are part of like biological systems pretty often for our materials because in theory they'll be more biologically compatible when you put them in the body. And I think a lot of skincare products have similar goals for like keeping skin healthy or whatever. They'll deliver things that are part of the extracellular matrix to uh, just provide some structure and prevent like degradation of skin things, I think. And so yeah, there is some crossover, which is kind of fun.
Yeah, it's kind of interesting to like see. I guess it is kind of the same thing is like the skin wound research versus the uh like the brain research. I guess it's kind of a similar idea, but it is funny to just see how much crossover there can be
Hmm.
in like a fundamental idea like that.
Yeah.
To kind of, land this plane, kind of bring it home. Um if somebody was like really curious about this kind of research or even just like tangentially related research, I think a really increasingly emerging field is like brain related research. Um, how do you encourage somebody who is interested but doesn't know where to go, doesn't know what to look for? Um, like they're interested in, like healing research, treatments for things, and maybe it's even brain specific, but how do they like get more information on this if they want it?
Yeah, great question. So, you know, one unfortunate part of academic research in general and like scientific stuff is accessibility is not always that high. I do have some advice for how to access things, but I think uh, we publish or we do some research. The way you get it out there is you publish the research in a journal, and um, journals they are companies, and so they put paywalls on top of all their stuff. The lab has to pay them a lot of money to publish stuff in their journal. Like it's a whole thing, and so it can be difficult to find papers sometimes. I think the best way um, to find information, I think lab websites tend to do a good job of describing what they do. And so, like, uh, if you were to Google a particular, honestly, like with the AI chatbots we have now, if you're interested in, like, hey, I wonder what kind of like, which labs do, uh, like, research on neural regeneration. If you were to put that in ChatGPT, which labs do research on neural regeneration? They would give you some names. You could go to their website to kind of read about what they're doing. Um, you can go from there to find some of their papers. If you ever use Google Scholar, scholar.google.com, you can just like paste their name in there. They'll have a profile probably, and you can see all of their papers. Uh, I don't know because you can have your papers open source. I think you have to pay the journal extra to make uh, your paper open source. I think that's the way it works. I mean, I don't know if there's a way to filter by like which papers are open source, but I think that's a good way to do it to be able to read some stuff. Um, and I find whenever I read papers outside of my own field that even as someone in like this like academia researchy space, it's always really hard to understand things because these papers always use a lot of jargon, um, you know, shorten things, do these things, and their argument would be that we're doing that because it's just quicker to talk between people in our own fields. We have these things and we know what they are because a lot of these names get really long. Um, Obvious con is that then if you aren't already in the field, it's like really hard to. understand what's going on. But I would just encourage the again AI stuff. I think can help with that. If you find an open source paper that you're like that title looks cool, I've started to read it and it's like really heavy, jargon heavy. You can download it as a PDF, put it into a thing, and say, hey, can you summarize this for me? And it will in a much more approachable way, probably. So that's probably how I would encourage getting some more exposure to these things, because there are, you know, media outlets. They'll often take time to publish, like, or not necessarily publish, but just talk about recent scientific discoveries, which is good. Like, I think it's awesome that they have an interest in showing scientific stuff. Um, it's pretty common for things to get sensationalized, like with lots of things in the media. Science is the same, and so like there will be a lab who there was one recently. They 3D printed like lab grown meat. Like they 3D printed some like meat type of thing. And if you read the paper, they'll say like we 3D printed this thing. It's it's an innovation because of this. It needs this before it becomes like a real thing. Like it's still in progress, whatever. And then a media outlet, just saying, "Oh my God, they printed live grown meat," which is very cool. But they'll take it and then they'll write the article. And if you read it, you would think, okay, like in a few months, we're going to have lab grown meat on our shelves and like we're ready to go. And so that's a more harmless example, but there are less harmless examples where that can happen. So if you do see a media article talking about something cool, that's awesome. They'll usually have the paper in there somewhere. Um, do that thing with the paper to make sure that what you're reading in the article is exactly what the paper's saying. You could probably even ask I think, hey, is this article like blowing this out of proportion a little bit, or is it right on point?
Hmm. Since you mentioned it, I will ask,
Hmm?
what do you think the role, if there is one, of AI or maybe even LLMs specifically in the research that you're interested in?
Yeah. So, think about biomaterial stuff. I mean, in biomedical engineering in general, AI is big, just like it is everywhere. I think, like in my building, there are like 3 or 4 labs that are like pretty AI centric. Um, they often do things like I mentioned that drug discovery research earlier, where like a big company will have like a million things and then like cycle through a million things to find which drug matches the target they're going for. A lot of AI is trying to do that in a simulation, and so instead of having to spend a lot of resources and time going through a million potential drug formulations, like maybe an AI model can give you like I don't know the numbers, but like I don't know 100 or 1,000, like much less. I think in our field, the you could in theory have like different biomaterial formulations, and an AI thing kind of tell you what's most ideal. Uh, I think in research in general, AI is making things faster, like just like it is in other fields. Just even with like lab organization and lab logistic things. Um, but yeah, it's everywhere. I think uh, a lot of people will say that they treat it as a tool, not like a replacement for like every people talk about like, maybe physicians will be replaced by AI because and AI think can like diagnose people probably pretty well. Um, but I think the prevailing thing among the AI people in biomedical engineering, who they would probably be the ones to displace physicians if someone was going to do that. Um, they just say that doctors who don't know how to use AI and can't use it or refuse to use it, they will be replaced by doctors who can and who do use it.
Mm.
So that's kind of the distinction they say, because uh, it's a tool and it's here and it's has a lot of pros and cons to it probably.
That's a great nuance. Kind of seeing it as a tool, as something that's an ability. Yeah. Jeremy Thomas, ladies and gentlemen, dude, you're awesome. This stuff is so interesting. And I think it's really cool that you're you're diving into this kind of treatment and this research to improve people's lives. I was very encouraged by like your why behind it. I think that's it's really admirable. Trying to help people who have experienced a really traumatic event that could affect them for the rest of their lives, at least try to like ease that for them. And if anything, I don't know, it's just seeing this side of kind of like the leading edge of science is exciting because like this conversation we're having with the AIs and LLMs, it can be a little scary sometimes. And so this is phenomenal stuff. It's so interesting. The nuances of the different types of hydrogels and the crossover between research and then how there's financial stuff behind those motivations. It's all really fascinating. And yeah, I really appreciate you coming and talking to me about this and kind of expanding my knowledge. And I'm excited to see as you progress us having some more conversations about how these research opportunities are growing and just the stuff that you're learning in this process. So yeah, thank you, man. I really appreciate it. Would you mind just telling people where they can find the research you're working on, what your lab is? Obviously, I'll link links to the website and everything, but tell people about that.
For sure. Well, first, thanks, Austin. You're great. Happy to chat. Happy to chat with you always. Yeah, let me make sure I get the right URL. Yeah, seguralab.duke.edu. That's our lab. Tatiana Segura is our professor leading our lab. And so if you Googled her, Tatiana Segura or Segura Lab or something, it would pop up. And we've, the like a couple years before I joined, they put a lot of work into the website, so it's a very like engaging. It's got a lot of cool stuff on it. So check it out if you're
Yeah, it's a
interested.
great website. Tatiana Segura, you said, and seguralab.duke.edu.
Hmm.
Awesome. Very
Yep.
cool. Again, I really appreciate you. You're amazing. And yeah, we'll do this again soon.
Sounds good.